Good morning, everyone. Welcome to our webinar. We'll give it just a few seconds to let everyone get connected.
Which means
hope everyone is having a good Wednesday morning.
Which means it's time for a little humor while people trickle in. So, I met the other day a real life microbiologist, and he was way bigger than I expected.
Okay.
So 3 noble gases walk into a bar. The bartender says, "Hey," oh no, it starts like this. Xenon, argon, and krypton walk into a bar. And the bartender says, "Hey, we don't serve noble gases here." The guys do not react. Okay, one more. So, of course, you're going to do introductions, but we've got a Lance on our panel today. You know, these days, Lance is a relatively rare name, but in the Middle Ages, people were called Lance a lot.
Yep, we can definitely see where this is at. Yep. All right, well, good morning, everyone. Thank you for joining our second of 3 webinars this summer. My name is Kinsey. I'm the U.S. Director of Sales here at Biome Makers. Today, as you can see, our title of the webinar is The Evolution of Soil Testing from Chemical to Biological to Predictive. We think we've got a great layout for the next 45 minutes to talk through. I'll let everybody introduce themselves. Scott, why don't you go ahead and introduce yourself?
Good morning, everybody. Thanks for being here. My name is Scott McElveen. I live in Burlington, North Carolina. I have a master's in ag science from Missouri State University, in which I weaseled my way into the microbiology department and used next generation DNA sequencing tools for my thesis. And I've been with Biome Makers for a year and a quarter at this point, and am just thrilled at the innovations that have taken place at this company to make biological testing effective, affordable, high resolution, and I'm thrilled for the opportunity to talk about it once more.
Awesome. Well, thanks, Scott. And we also have with us today Lance. Lance, you want to introduce yourself?
Yeah. First of all, thank you guys for having me. I really appreciate it. My name is Lance Gunderson, president and co-owner of Regen Ag Lab in Pleasanton, Nebraska. I've been involved with soil testing for around 22, 23 years now. At Regen Ag Lab, we're really focused on kind of moving beyond the traditional approach of soil testing, which obviously is why I'm kind of part of this webinar. That's what we're going to talk a lot about today. I got my PhD at the University of Lincoln in Nebraska in soil microbial ecology. I am not a traditional soil scientist, but that's all right. I eat, breathe, and live it every day. So we're going to dive into it. Thanks again for having me.
Thanks for being here.
Well, let's get into next slide, Scott, of what we're going to be talking about today. So goals of the webinar, brief history of soil sampling. You might be asked why now. I think it should be fairly obvious. We're towards the end of the season, we're going into fall, and so we may be looking at fall soil sampling, resampling, maybe looking at what new technology is out there. How do I pair this with something that I've already done? So what are the options? And it's not just about understanding the options, but why and how those options can be beneficial to you and your operation. So we'll run through a brief history of soil sampling and some of the chemistry or things that have been involved, along with some of the new tools. And Scott's going to run through some of the stuff when we get to it around how important is new technology. So being able to be predictive, like AI and machine learning. What's that done for us, and where do we think we're going? And then, of course, we'll end with a Q&A session and engaging with the audience. So with that, Scott, I think you're up to get us started at the moment.
Off we go. We promised you a history. Broadly understood, soil testing is old as dirt. So the 5 senses, as long as there's been agriculture, humans have been looking at soil and understanding differences in color and taste and aggregation. I mean, tasting just for salts, feel. You can learn a whole lot from just 5 senses. But the specificity, accuracy, and precision of our technologies for characterizing soil and its manifold properties have undergone a continuously accelerating development since the late 1800s. Soil, of course, can be broken down conceptually into biological, physical, and chemical components and properties. The physical properties are essentially important. We have all kinds of tests for physical properties in soil that I think get undervalued because they're so accessible in DIY that we overlook them. But basic ribbon tests for understanding aspects of soil texture, a basic soil infiltration test, watching how quickly can my soil take in water relevant to, of course, your crop hydration, but to runoff and erosion, and how your soil can deal with a 1-inch rain event, a 3-inch rain event. Got the classic Munsell color charts. If you've ever been through a Soil Lab 101, you have done color, I guess it's not, maybe it's not colorimetric, might not be the right word, but you've analyzed the hue and the value and the chroma of different soils to type them. A slake test can tell you so much about your soil's water aggregate stability. And so all of these soil physical tests are a great topic for a different webinar, but I think they deserve a shout out to remind us going into fall, there are things that I can go out and do to better understand my soils and ways I stand to gain from improving soil health and function. Now on the chemical side, I'm about to hand things off to resident expert, Dr. Lance. As far back as Justus von Liebig, he was using hydrochloric acid to extract nutrients from soil, understanding soil, basically that we could extract and identify the nutrients that would be plant available or unlockable in soil. Today, we have all seen and are familiar with the way that we have understood how pH influences which nutrients are available and unavailable. And to this day, Justus's sets of, I don't even know what to call this chemistry contraption here, has evolved into inductively coupled plasma following extracts that have become prevailing in use in modern contexts. So Lance, will you tell us about the development of extracts for soil nutrients?
Yeah, sure, Scott. Man, that's an old ICP 7000 from Thermo. We've run a couple of those. So yeah, the technology on the instrumentation is progressing quickly as well. You know, when soil testing first started, kind of in a commercial sense, if you will, it all kind of started right around the end of World War II. And we were focused on the soil extract. And when we talk about an extract, what we mean is when you send a soil off to a laboratory, different chemistry is added to it, essentially to solubilize or pull out what they call plant available nutrients. And there has been argument since 1940 on, you know, which extract should be used where, what nutrients are really truly available, are they really available to the plant, et cetera, et cetera. And that is a long running argument, and it will continue probably until I'm long gone. But the idea was to focus on this extract to develop one that we could use on various soil types. So in 1945, Bray and Kurtz published Bray P1, or sometimes referred to as weak Bray, and they used that primarily for phosphorus determination. Still a popular extract today in certain states. The weakness of it, though, is that it didn't work well in calcareous soils. It worked well in relatively low organic matter soils, lighter textured, lower pH. So on the eastern third of the country, and especially the southeast and in those regions. Now, I know you can argue, yeah, well, you know, it's been calibrated here and calibrated there in different states, and you know that's fine. And yes, that work has been done. But the argument is still, you know, how did we draw state boundaries to where an extract works within a certain state boundary, but not another state boundary? So again, that's where the arguments kind of have come from, and it's come from the research, different universities at different states. And so in 1954, Olson developed the sodium bicarbonate extraction, or the Olson extraction. This was centered around phosphorus determination again, but in high pH calcareous soils. So out in the Western Plains and west of the Continental Divide, that is a very popular extract. Both of these were limited though, because they only looked at really phosphorus. And so Malik set out in 1953, you can see this is a long journey for Malik. In 1953, was working on a multi-nutrient extractant. The idea was to use a single extractant to measure phosphorus, potassium, calcium, etc. The biggest issue, well, so Malik 1, I guess, was introduced in 1953. It actually is still used today in a couple of states. Malik 2 was introduced in 1978. Not knocking on Malik on this one, but Malik 2 was kind of a flop. It wasn't adopted. Nobody used it much. 1984, though, he hit a home run for his extract for his work, and Malik 3 was published. And since then, Malik 3 has been the predominant soil extract used in the soil testing market in the United States. One of the biggest issues with Malik 3 is that, again, the argument's still there that, well, it doesn't work really well on calcareous soils, high pH soils. It overestimates P and phosphorus availability, or excuse me, phosphorus availability, because it extracts it off of calcium. But one of the other big headaches of it is that Malik 3 is made up predominantly of ammonium nitrate. You cannot test for nitrate and ammonium with an extract that is made up of ammonium nitrate. It is the reason why the standard soil test east of the Mississippi was pH PK. Nitrogen was not included because everyone was using Malik 3. So in 1981, Whitehead published the KCl extract method, and the KCl extract method then is for nitrate and ammonium. Nitrate is soluble in water. So the KCl was not necessarily for the nitrate portion. It was a dual extraction, and the KCl was used for the ammonium. Ammonium is fairly soluble, but it is a positively charged ion. So in high CEC soils, you can, and if you have a high ammonium load in the soil after fertilization, you will have ammonium on the CEC complex. So the KCl helps exchange the ammonium, and that's what the KCl was used for. Again, this really did progress agriculture. Keep in mind, in the 1940s, what was being developed, in the 1940s and 50s, and that was fertilizer. The fertilizer industry was kind of being born after World War II, and these tests were supposed to help producers understand what they had in their soil from a fertility standpoint, and then could make decisions on what to apply. They are limited though. Again, arguing about which extract should be used where. The other limitation is that all this chemistry, this benchtop chemistry, it really overpowers the natural soil system. So moving on to the next slide, we're going to talk a little bit about the evolution of extractant. So Dr. Haney, when he was going to school, he went to school to be a soil chemist. He wanted to kind of follow in the footsteps of Malik and develop a multi-nutrient extractant, but he had two major goals. And the first goal was, I want an extract that is not going to overpower the natural soil environment. That was the first goal. I do not want to overdrive the pH. Number two, I want an extractant that will include nitrate and ammonium, so we can have a single extractant that really does capture the nitrate and ammonium portion of this. And I guess kind of a third goal was, what it kind of evolved into was, I don't want to use any chemistries that are not naturally occurring in the soil space. And I'm not saying we're using, the other guy's using synthetic things. I don't like that label, synthetic and non-synthetic, but he meant things that are naturally occurring in that environment. So in 2006, he had done a ton of research, literature review, and said, okay, I want to develop this extract. I'm going to use some organic acids that plants produce, And plants produce hundreds of these different compounds. They leak them out into the soil through the roots. But the research showed that there were 3 or 4 predominant organic acids that were produced during the growing season in most of our agriculture crops, especially during times of high nutrient uptake need. So he paired those into an extract. So 2006, he published on H3A1, version 1. H3A just stands for the 4 people who worked on this extract. It's Haney, Harmel, and Arnold. That's a pretty tough mouthful, so condense it down to H3A. The first thing he did is he compared it to Malic 3 and 1 normal KCl and published that work. He was showing the correlations of this, how well does it line up with these in different situations. It needed some work, so he continued working on it. In 2012, he came out with H3A version 2. He removed the EDTA and the DTPA that were in the extract. Those are chelators. They're commonly used in most of our conventional extracts. Matter of fact, we just have an extract called DTPA, or micronutrients. And he was using lithium citrate as a buffer against pH change on his extract, and he reduced the amount of that. 2016, slightly modified version 3. All he did there, or version 2, all he did there was change the concentration of the organic acids a little bit. And then quickly after that, in 2017, he removed lithium citrate completely from the extract, and his reasoning there was that lithium citrate does not exist in a natural soil system. Plants are not making it, microbes are not making it. So these are the things he was working on, and he worked on correlating them to conventional extracts. So on the next slide, we will show you, this is some of the work he did originally. Again, I said one of his primary goals was to not overpower the soil pH. So what we're looking at here is he collected soils, or had soils sent to the USDA ARS research lab in Temple, and they varied in pH from 4.5 all the way up to 8.5, 9. And he had these soils sent in from all over the country, and he took those soils and he extracted them with some of the popular, a couple of the most popular extracts, Malic 3 and Olson. And what he did after he extracted it is he took the liquid that he used after being exposed to soil, and he pH'd that liquid, so the filtrate. And the pH of the filtrate on every soil extracted with Malic 3 fell between around 3 and 4. I think 4.2, 4.3 was the highest one. Malic 3 has a pH of around 2.5. And it's very strong. It's not just because the pH is low, it's a strong acid. It is highly buffered, which means it can overpower the soil pH. So a pH soil of 9, the extract gets driven down to a pH of 4. And he said, okay, he's doing this for an illustrative purpose. He goes, okay, well, let's do Olson. He took all the same soil samples, he extracted them with Olson. The resultant pH, Olson has got a very high pH. Don't quote me on it. I want to say it's around 13. Mark, do you know? I think it's right around 13 on that bicarb. I should have known that. But we don't.
Yeah,
But
I think you're right.
High. Yeah, and so again, same idea, right? Sodium bicarbonate extracts the soils. Well, guess what? The pH of the soil that was 4, 5, 6. They got driven all the way up to a pH of between 8.5 and 9. So he tried water. You say, well, it rains water. You know, let's look at water. So water has very little to no buffering capacity, right? Pure water. He used deionized water and said, okay, well, the resulting extract followed the soil pH. You know, you see the lowest one is around 5.2 and the highest one's up in the 8.5, 9. Mm-hmm. It follows the soil pH, but water itself doesn't have a lot of extracting ability. And what he's looking at here is phosphorus. I should have mentioned that on the y-axis. but he's looking at phosphorus. It doesn't have a very good ability to extract phosphorus. Well, it rains water, but water mixes with soil, and soil contains these naturally occurring acids, right? So then here with H3A, we've got a cluster. So we've got a cluster of pH. So the pH of H3A is about 4.5. It's lightly buffered. It actually smells like Tang, for any of you old enough to Tang, the astronaut drink. It's made up of citric acid, oxalic acid, and malic acid. And if you look at the back of almost any food label, you're going to see those three acids, at least one of them. So acidic to slightly acidic to neutral soils, they kind of clustered around 5 to 5.5 for a pH. There's a couple of them right around 7, and then there's a few of them in that bottom right corner at 7.5 up to 8.5. So your calcareous soils, they came back with a higher pH after the extract, and your neutral to acidic soils came back with a slightly acidic pH. So this was his first step. Now you could say, well, that's great, but what does it have to do? What do the numbers mean? But remember, his first goal was just to say, I want an extract that will not overpower the natural soil pH. So moving on, he said, okay, here's my second version of this. We need to see how well this correlates to conventional extracts. Now, the funny thing about correlations is, so when I show you these R² values on the right-hand side of this slide, the closer that value is to 1, the better the correlation. Correlation, though, does not mean that you get the same number. So it does not mean that ammonium acetate potassium and H3A potassium on the same soil give you the exact same result. So if ammonium acetate is 200, that does not, part per million, that does not mean H3A is 200. What it means is that when you take the two lines, if you run a bunch of soils and you take the two lines and lay them next to each other, they are in a very good relationship. They might have different slopes, but they have a very solid predictive relationship. And so you can create a conversion factor with a 95% certainty to convert from one number to the other. Now, this is the part where everybody kind of lost their minds and said, you can't do that. And I said, well, we've been doing that in soil science for 50 years. There's probably a number of you on this webinar right now that can tell me the conversion factor from Bray P1 to Malic 3P, or Olson to Bray. So that is exactly what they've been doing. So we can see here, correlation coefficient, the ammonium acetate was the kind of tried and true extractant. I know Malic 3 has replaced that in a lot of labs, but again, ammonium acetate for potassium was kind of it. There's your 0.95 KCl. Nitrate is soluble in water, so again, the extract doesn't matter as much. 0.97, we get to Olson at a 0.82, Malic 3. Phosphate, that's colorimetric, and we'll talk a little bit about that in more detail in a couple slides. But the colorimetric Malic 3 is 0.81, ICP is 0.8, calcium for Malic 3, calcium for, you can see those correlations. Now, some of you might be saying, well, what about zinc? Or zinc's on there, excuse me. What about iron? You know, what about copper? We have correlations for those. Rick didn't put those in this presentation or on this slide that he did because he originally was not looking at those. He was focused on NPK, calcium, some of the micronutrients he was not including at the time. Since then, we have done that. We've done the correlations. Not all of them are this strong. Copper, it's a very difficult correlation. Copper is such a small number that you can run the same sample in the lab 50 times and you go, you know, 0.16, 0.18. Well, 0.16 to 0.18 is a large percent difference because that number is so small. nitrate at 12.2 parts per million and 12.1 parts per million is technically a larger absolute difference, but it's a very small relative difference. And so that tightens up a little bit of the numbers. So not all of those work as well, but we didn't just highlight the ones that did. These are the ones Rick was working on. Next slide, please. So the next thing he did is he said, okay, well, if most of these different extracts exist because of phosphorus, I'm going to look at this and I'm going to go back to one of the oldest methods that most of these were kind of gauged against, and it's iron aluminum oxide strips. No commercial lab runs iron aluminum oxide strips for phosphorus. I shouldn't say that. I don't know. I haven't visited all of them. I have never heard anyone say they use iron aluminum oxide strips anymore. I don't believe you can commercially buy them. You have to make them yourself. Rick did make these, but this was the tried and true. So he took iron aluminum oxide strips, tested the phosphorus. He extracted that same soil with H3A. This was version 3. Tested that against phosphorus, and he got a 1-to-1 relationship in this case. Very close to it. The slope is 0.95. A slope of 1 would be 1-to-1. A very small y-intercept, correlation coefficient of 0.98. Again, 1 is perfect. If you get above 0.5 in soil science, you're on the right track. So this is what he's showing. This has been, correlated back to the original tried and true. Next slide, please. There was one big problem between H3A version 3 and H3A version 4. The extracts were cloudy. Now, cloudy extracts, it doesn't matter when you're running ICP, unless they're really, really cloudy. But cloudy extracts for ICP don't matter as much. ICP, they're going to burn the sample. Every element gives off a certain wavelength of light. The instrument measures those different wavelengths of light. How intense the light is relative to concentration. When you do colorimetric, you're actually mixing different color development reagents to the sample, and you're going to develop a color and you're going to shine light through it and you're going to measure absorbance. Well, cloudy samples give you false absorbance. And so for things like phosphate or ammonium, if your color development or the light you're looking for is close to the color of the cloudiness in the sample, you get a false positive. So it looks like you have a bunch of ammonium in your sample, but you may not have much at all. So his goal then was to, how do we remove this lithium citrate and clear up these samples? So he's just showing you a comparison between two soils where they were both extracted. On the left, you have the same sample extracted with H3A and H3A4, showing that cloudiness. And then he's got another sample showing you that as well. This was really a problem that became more apparent when this test was adopted commercially, because different labs have different instrument platforms, and they might all measure the same thing, but they have different requirements or things like that that are going to place constraints. And that's exactly what happened when I started calling him saying, how come our numbers don't match yours? They match yours on everything but these 2 or 3 factors. And we figured out that it was cloudy. He went back to the lab, figured out how to remove that. Of course, went back and retested how that affected phosphorus correlation, how it affected nitrate correlation, and it did not. And so since that time, since 2017, we've been running the H3A version 4. So that's kind of the evolution and the steps. Now, again, this extract, this piece of the extract that we talked about is still just focused on chemistry, right? It's still just measuring fertility in the soil. So this is not the Haney test. This is just what became part of the Haney test that we'll probably talk about a little bit here in more detail.
Yes.
Yeah, Lance, thank you for going through kind of like the evolution of chemistry. It is exciting to see a relatively new extract and methodology, and we'll get into the benefits and really what the Haney delivers. But thank you for the background, the chemistry lesson, if you will. Greatly appreciate that. So I think Scott, you're moving into the next phase, correct? All right.
Will. Lance, will there next year be a 20-year anniversary celebration of H3A1?
You know what? We hadn't thought about it, but we probably should. We probably should have a party. Might be a short guest list though. It's like anything in soil science or anywhere. You know, there's always going to be there's a lot of pushback, right? There's a lot of pushback, and that's okay because that's how science progresses. And
yes.
So there needs to be pushback, and then there needs to be positive pushback, meaning these are the things that are wrong, and here's why, and here's how we're going to move forward, right, rather than this is why it's wrong and we're going to move backwards. And that's really the discussion that I've been having for years, to say, well, we've been doing it this way for 40 years and we're still having the same discussions, the same problems. I'm still getting the same things from growers. You know, I put on the P, I put on the K, where is it? How come I can't get it? My plant's still showing deficiency. And that's really the reason we're all here having this discussion, to say, well, what can we do that's different and not always right, but doing it wrong the same way every time is not helping either. So again, those extracts have gotten us a long ways in agriculture. There's no argument around that.
Oh, totally agree.
Yeah, they have certainly helped us, and I'm not dogging on any of them. It's just, how can we make this? What's the next step?
Yeah, I think it's a great foundation to build off of, and we'll definitely get to that here in probably another 10, maybe 15, 12 minutes. So.
Okay. Well, before we circle back to that, because we want to conclude with the discussion of why and how do we employ these solutions effectively, we got to talk about the biological end of things. Y'all expected it. It's a Biome Makers hosted webinar. Studying soil biology, we're talking microorganisms, macroorganisms, and the plants themselves. And so, I mean, Charles Darwin's last book was a study on the vegetable molds produced by earthworms. Earthworm counts are as old as dirt. But microbiology, soil biology changed a lot with the development of the first compound microscopes, where it was suddenly discovered there was a whole lot more, there were many more living organisms than we were aware of, because we could not see them. Soil was not simply dead particles to hold plants up while water and sunlight and manure nourished them. No, now we are able to clearly see this is a predatory nematode that was from my own backyard last year. You can do these sorts of images just at home at your desk. And then we have higher clear scanning electron microscope images where we can actually observe nematodes interact with tiny microarthropods. Also, there are cell culture approaches to understanding the organisms that are too small to characterize well with common microscopy tools. Overwhelmingly, your nutrient cyclers, your organisms that enable plants to deal with stressors, the majority of those that cause diseases, they are too small to characterize and differentiate well with microscopes. And so one way to approach that is to culture them in a petri dish, get them to grow in a lab where you can subject them to different tests and characterize them. How do they respond to this or that stimulus, this and that substrate? The trouble with cell culture is soil is a highly diverse set of environments, whereas a petri dish in a laboratory is a very narrow set of conditions. So you're only going to capture the organisms that like that pH and that level of light exposure and those nutrients on the plate and that level of oxygenation, and you capture a small sliver of what's living in the soil. It's useful sometimes because we identify specific pathogens that we know if we set them in these situations, and we apply a soil sample, we can get it to grow, find out if you have that pathogen present. Same goes for things like bradyrhizobium. But when it comes to characterizing the whole soil ecosystem and the world as the crop sees it, cell culture is pretty much not useful outside of specific research settings. And so soil DNA testing provided a way to capture the broad swath of organisms, microorganisms in soil. Here we have a rotating 16S gene. This is the gene that we at Biome Makers pull basically a DNA fingerprint from to identify what bacteria and archaea are in a sample. There's a separate gene, ITS1, that we use for fungi. I'm not going to belabor how bee crop works because we've covered that time and time again. I'm going to briefly remind you, we extract these these marker genes from organisms, identify who they came from with the help of a database that took many years, lots of sampling, lots of collaboration, lots of investment to build. And then the first big breakthrough for bee crop was the ability to not just hand you a list of indecipherable Latin names of organisms and how many are there, but rather to class them into what functions they perform so that we can give you, here are your nitrogen mineralizers, your phosphorus solubilizers, your pythiums, as well as your organisms that suppress pythium, so on and so forth. Now, if many of you watching this webinar are already familiar with that, what we want to talk about today is the problem of spatial variability. How then, I've taken this tiny sample and sent it to you, how are you going to characterize my farm, my landscape? I do that through chemistry using grid sampling, or I've got different zones that I use to tackle the variability across my landscape. But gosh, how am I going to use bee crop tests that cost more than my soil fertility tests and yet capture variability across my landscape, the high spots, the low spots, the wet spots, the dry spots, so on and so forth? And so the way we have unlocked that capacity is we take the fields, we take shape files from those fields. Those are uploaded to the bee crop portal. Then that landscape is analyzed on the basis of the relevant spectral and environmental data that influence microbiome composition and function. We were able to unpack this. This couldn't have been done before because it took time to build a database of samples that were georeferenced and had dates to where we could assess the relationships and what were the important drivers between environmental and crop response data and differences we would measure in microbiome composition and function. So through the spectral analysis, we can differentiate the properties, the characteristics of each 10 meter by 10 meter squared section of soil, cluster the pieces of the landscape into groups of like soil with like as far as the relevant the properties that drive biological differences. And then with that, we select optimal coordinates and sample locations, send those to the person who will be pulling the samples. Now, if that was a little, like for me, if I'm just seeing that, that's a little bit over my head. And so I had to think this through probably 27 times before I really grasped what it is. Y'all may be smarter than me, but here's my analogy that helped me get it on an intuitive level. So let's say my machine learning algorithm, my clustering algorithm, this is a machine learning process that has been in development for decades, used for decades. It's not the sorts of AI that are our current large language models that we can't really see into. This is a statistical tool that does something akin to the following. I give it a huge pile of movies, and I say, I want you to cluster these on the basis of their characteristics. So I'll tell the algorithm what it should look at. I'll say, I want you to look at the story beats, the predictable tropes. I want you to look at characters, casting, tone, lighting, the score, sound dynamics, pacing, word choices, and then just give me 5 different buckets of like with like. And what that algorithm is going to do, I don't have to tell it, this is what horror movies are like, this is what action movies are like. It's going to put Mission Impossible, James Bond, and the Bourne movies in a group together because of the commonalities they share of espionage and assassins, and even the music is similar. It's going to put Will Ferrell, Adam Sandler, and Kevin Hart movies into a bucket. It's going to put Nightmare on Elm Street, Halloween, and Scream into a bucket because they are similar, because they have commonalities. And it's going to give me a collection of clusters. Now, our movies in this analogy are our 10 meter by 10 meter square bits of land. Now, say each of those movies is a dot, all the algorithm is doing is it's clustering them into groups of like with like. That's as complex as it is, except that instead of just having 2 axes, we've got 1,000 axes. It's in multidimensional space. So you've got to think about your soil properties being, well, more spread out than just along 2 axes, or even the ones I said for the movie. Our movies, I gave you like 12 different dimensions by which we analyze those. So each bit of land is a movie. Those bits are grouped into clusters with similar properties. We then strategically target samples to ensure that each zone is represented in proportion to its size. So some characteristics don't matter. Having a title card will not separate one movie group from another. Having end credits won't help to differentiate movies. We had to learn what properties drive microbiome composition and function so that we could train the clustering algorithm to pay attention to what's important. But now that we can do this, it allows us to characterize a landscape at an affordable price. This is, I don't know, I'm a business development rep, so you got to, you know, I know that this is coming from a biased source, but even before I joined Biome Makers, I was convinced that breakthroughs like these were really groundbreaking enablers for understanding soil biology, nutrient cycling, disease risk, stress across an entire farm. This could not have been done before last year. We're approaching, you know, Bee Crop Farm's first birthday.
Yep.
This is a new frontier in finally soil biology, high resolution, clear metrics being accessible to
So Scott,
commercial farming.
I think it's worth mentioning how many layers, like you think about data, you think about, for those that have been on the call and worked with spatial data before, and you think about processing yield maps or trying to run a few equations that reference 2 or 3 maps, and you know, some of the old computers, how long that took to run. Scott, how many layers or how much data actually goes into the machine learning or the our way of setting sample points?
There are over 1,000 layers, parameters that go into this process. They're weighted differently based on their importance, but yeah, to your point, computing power would not have been adequate to affordably do this, what, a half decade ago? I'm not an expert on computing power advances, but I know it advances rapidly.
Yeah, I think it's testament to what Lance talked about of, you know, from a chemistry side, you had, you know, Malic over how many years to bring Malic 1 to Malic 3. We've seen, you know, the advancements, and we'll talk more about what the Haney test does. And then, you know, we started out with bee crop tests and being able to give samples and actionable insights at a point value. And now then today, through all the advancements, we're actually able to spatially lay out results and gather insights that can now be driven, you know, at a subfield level. You know, now can be interacted with, you know, with some of the advanced sprayers, the planter technology, drones. There's a whole host of ways of being able to use this technology now that we haven't had in the past.
So if you're at the stage of Bee Crop Farm where you've got to upload shape files, and what are they doing with this? The whole point is we're looking for the impassioned Jimmy Stewart speeches, the Michael Bay explosions, the rom-com meet cutes, the Morgan Freeman narration, all the things that help us predict before we even go out and sample. We're going to predict, here are the clusters, here's how the microbiome is going to shift and vary across this terrain. And now we're strategically sampling, which saves a great deal of resource and effort.
I think it's important to share, Scott, you know, there's a whole level of validation. So Lance, you talked about, you know, how this was validated in R-squared and stuff. And I think it's important to share that, you know, we've got several examples. I'm going to pick on my favorite one of when we first started doing this. We were looking, and a customer wanted 26 samples to represent their field, similar to what you see on the screen. And so we did that. We gathered the 26 samples with the points. We also ran using the AI model to pull 11 points. And so both of those were run through the system independently to generate the maps and results. And then there was the difference map, the delta was looked at, and as far as were we able to, from a predictive standpoint, utilize this methodology and then use our technology to get to that answer. And the short answer is yes, we were 98% accurate of being able to use our smart zonification over a more intensive sampling method. So again, I share that just from the fun of sharing and excitement of what the technology does, but also from a confidence standpoint of how far has all this technology that we've talked about from soil testing has come over the last several years, and how I believe it's going to continue to grow. So
that's the predictive bit of the webinar title, and now we come to the question of, okay, but why? Crop prices are in the pits. How can these tools offer value, offer better outcomes over the program I've already been doing? And maybe we start with Lance for this.
Yeah, Lance, let's. You gave us the chemistry behind the Haney test, but really, what is the power in the Haney test, and what's the advantage, and what does that do? How can that complement somebody that's already maybe thinking about pulling they're on a new set of cycle for the year, or maybe they've got samples that are still good, but they're looking to find something new. So can you elaborate on what the Haney test is going to do for us this fall from a knowledge and decision basis?
Yeah, and I might approach this a little differently, but I'm an analogy person, if you haven't heard me speak before. So the analogy that kind of hit me was, 5 years ago, I'm trying to start a lab, and we went through this thing called COVID. And I remember sitting here going, okay, look, I'm trying to buy this. I'm trying to get these supplies. Everybody I call says we don't have it, can't get it. I was like, well, is it because they're not making it, or what's the issue? And what I found out was it was a distribution problem, right? So there were container ships parked out in the harbors full of stuff, gloves. You couldn't get gloves to work in the lab. You couldn't get test tubes. You couldn't get anything. And it's like all this stuff was sitting on container ships. You had demand, you had empty store shelves, and we couldn't get distribution. And so the analogy here is that we have done a very good job in agriculture about looking at what is in the warehouse. That's your soil test. Say, well, what's the soil warehouse have in it? And you could say, yeah, I can measure total nitrogen, and that's everything in the warehouse for nitrogen. I measure nitrate, and I say that's the stuff on the bottom shelf that I can get really easily. And then we'll evaluate the plant, and that's the factory. And you say, well, evaluate the plant. Am I getting the right stuff in the warehouse? But if the answer was no, the solution was always go back to the warehouse and put more in the warehouse, right? Like if my plant was short on phosphorus, the answer was always, well, let's go back and put more P on the soil. And we've done that for 50 years. And in some cases, yeah, if your warehouse doesn't have any phosphorus in it, you need to put phosphorus on, right? But that hasn't been the answer. And we've got so many people out there saying, well, I don't have this available. So one of the things with the Haney test is, well, maybe we've been looking at phosphorus with the wrong extract. And I'm not saying it's a wrong extract in every situation. I'm saying that maybe we're using the wrong extract in your situation. Maybe we're not truly evaluating what the plant actually has access to. What is truly out there that can be solubilized? Maleic 3 potassium can be 400, 450 parts per million. Ammonium acetate on the same soil is 200 to 250 parts per million, and H3A is going to come back at 100 parts per million. Which one of those numbers truly represents what's available? That's the argument. It's still the argument.
Sure.
So that's one of. Yeah, go ahead.
That actually, if I can interject with the first question that came up, it is perfectly attached to what you're saying now. And maybe you can work it into your answer. He says, or he or she asks, if H3A is so highly correlated with other testing methods, then what's the advantage of H3A over the others? If they're so well correlated, it seems like you'd end up with a similar decision at the end of the day regarding fertility.
They're correlated, but they don't give you the same number. So for example, the way all the original extracts were developed and then the way they built recommendations was they kind of developed these tables, and we've all seen them. You know, from 0 to 5 is very low, 5 to 10 is low, 10 to 20 is moderate. Well, that's true with that extract, but if they're correlated, that doesn't mean you're getting the same number. And so, for example, I can go out to Montana and run zinc. The zinc number comes back at 3 parts per million. According to that table, zinc is not low, but the crop is exhibiting zinc deficiency. Well, when you run H3A, the zinc comes back at 0.3 because the pH of that soil is 8.3. And so we're overestimating true availability with that higher acid extract, that stronger extract. So the correlation is there, the relationship is there. The difference between the two numbers is a pretty static number. But when you put both of those numbers and compare them to an agronomy table, you would say, oh, well, I don't need zinc. I don't have a problem with zinc. Why is my plant showing zinc deficiency? It shouldn't. I'm just going to put more zinc on. And then over here, you're like, but no, I do have zinc deficiency. And I put this number on a table, and I'm seeing now that this is low. It has a lot to do with, yeah, they're correlated. And they're correlated for a reason, because you have to be able to compare, in this case, apples to oranges through the correlation to make sense of it. Again, the foundational piece of the extracts are there, but we run into those situations all the time. And now outside of the extremes, yeah, I don't care what extract you use. If your phosphorus is 3 parts per million, that's very low. It doesn't matter. And it doesn't matter what crop you're growing. If it's above 70 parts per million, it's high. It doesn't matter. It's in the agronomic range that really makes a difference, because I know so many people that will go out and they'll say, well, my soil test says I've got 20 parts per million phosphorus. I put phosphorus on, saw no response. And the neighbor goes, well, mine was 20 parts per million. I put it on and did get a response. And that's where that second piece, that has nothing to do with the extract. That has to do with the next piece, which is distribution. Mobilization. So that's where the biology piece comes in. And we're seeing that more and more and more. 10 years ago, I could maybe name 4 or 5 farmers that were growing crops that made 0 sense. When you looked at their soil fertility numbers, it made no sense. Phosphorus 12 parts per million, Malic 3 extract, and they're generating 200 bushels of corn. And everyone goes, well, that's not possible. Well, I've been doing it for 10 years. So the next piece of this, the evolution of this, is saying, well, how do you look at the biology as a distribution? Your forklift operators, your truck drivers, your dock unloaders, those are the microbes that are moving things from your warehouse to your plant. But we want to look at the nutrients that these distributors actually have access to. And they're not generating Malic 3 to access zinc. The plant is feeding them organic acids. The organic acids temporarily change the soil pH on a micro scale. I mean, like the size of microbes, micro scale around those areas. The microbes use those as fuel, as food to do the work. The pH change helps mobilize some of these nutrients. And that is what they have access to. So the extract is designed to mimic that. It's to try to look at that better. Is it perfect? No.
So if I could attempt to, I'm going to try to say back to you what you said in different words, and you tell me if I've got it right. So you have witnessed, and the world has witnessed, anyone in farming probably has seen plenty of times where you have a crop response or lack of response to a fertilizer application, which if the soil chemistry lens is the only lens you have, is puzzling or is without a good explanation. But when you bring in the biological element, it is very likely the missing element for describing why fertilizer applied led to a response in one case and not another, or where no fertilizer was applied and you see a response that you shouldn't expect under the soil fertility testing alone paradigm, because okay, nothing went on, the levels seem too low, why is the crop doing fine? And it's because the microbes are the ones who are able to cycle nutrients from the warehouse, the locked up unavailable form, into the soluble available form via mineralization, solubilization. Am I tracking?
Yes, and transport. Dr. James White talks a lot about rhizophagy, right? And so these microbes accumulate nutrients, and those nutrients are stripped from the microbes through rhizophagy, or they're delivered through conduits such as AMF, mycorrhizal fungi. And so understanding that workforce in the biological world is that they are the gut of the system. And much like human medicine, yeah, you have to provide the right foods, but you're really providing the base level nutrients and energy for the microbes to do what they do in your small intestine. And they transport the nutrients into your system. Gut health, right? It's not just your small intestine just sucking everything up. That's what we think roots are doing, right? Just take it all up. So it's really this interaction between the two. The Haney test is limited in that it doesn't look at the level of, it's not measuring P solubilizers and potassium solubilizers or immobilizers. It's grouping this together as soil respiration as an over-encompassing look at life. The more your soil respires, the more life is in your soil. The one nutrient it looks at really hard is nitrogen, because these microbes are eating carbon and they're eating nitrogen, and they digest soluble protein first. That's what microbes want first. Look at all the digestion studies on ruminant animals. Who's doing the digestion in a ruminant animal? Microbes. What do they digest first? Soluble protein, followed by simple sugars, finally fats, and then finally fiber, right? The cellulose, hemicellulose, etc., lignin. So the microbes are going after that soluble protein, and from that, we do give an additional nitrogen credit from the Haney test. That was the big home run on Haney test, and it's because we saw growers all over the country that said, well, I'm using a much reduced nitrogen rate. I built myself up to that with my management practices. The next evolution in this step, though, is, well, what about zinc? What about boron? What about, and the Haney test doesn't look at all those. We can indirectly say, well, geez Louise, your soil respiration is 300 parts per million. You got all this carbon out here that is soluble. You've got this good fertility panel. That's great. So you're probably moving some of these things, but that's where things like B crop come in, because now you're able to look for and identify the organisms that we know through the database and have the function of creating these genes that move phosphorus. We know that these organisms produce the genes and functions to solubilize potassium.
But it's not just knowing that it's knowing where they're at in the fields and being able to map this out and being able to add this now as having a pretty good background in working with spatial layers and stuff. You'd find those similarities, and is it a landscape position? Is it, we were always looking for some layer where before we had, now we have B crop farm and the ability to look at these, at a subfield level of what's going on. And I think this is really going to, unlock the future of looking at what is driving yield. Yes, we've always tried to correlate it back to fertility levels. But it really kind of leads into, for lack of a better time, kind of leads into what our third and final webinar that Scott and I are going to delve into a week from now is getting into some of those nutrient cycles and how this all plays together. So how do we marry what has been traditional and is that foundation. But then how do we marry that with, Lance, I guess, we're right at time. So a couple last closing words of, what's the importance of marrying the Haney test with what they're doing, especially in a year like this where commodity prices and stuff can be a challenge. And then also, I'll give you a chance to throw in how B crop, and then Scott, I'll give you a word, and then we'll close this thing out, and we'll have to answer any remaining questions offline. So, yeah, Lance, I'll give you 30 seconds to 60 seconds to answer that. Sorry.
No, so the Haney test is going to evaluate what you got available in the warehouse, and it's going to give you an employee count. So it's going to say, this is what you've got, this is how many this is how big your workforce is, and now where do we go from there? You transition into B crop and say, well, okay, you've got 10,000 employees. Who are they? Because if all 10,000 of them are forklift operators,
we have a problem.
Yeah, right. So now it's looking at who they are and what they do, and it's the what they do that's important, more important than who they are. So then what they do, and that's how those two things pair together, because you can have all the P solubilizers out there you want, but if you don't have any phosphorus or very little phosphorus even available to start with, so you got to see both. And it's not about eliminating the fertility, it's about placing fertility where it's needed and then utilizing it properly.
Awesome. Thank you for that, Lance. Scott, anything you'd like to add?
Well, of course, that presentation Lance just gave right there about B crop, that's been true for the last 10 years. But the trouble was the spatial variability issue. And so if anyone hasn't heard about B crop farm, the spatial variability issue has been dealt with to where now we can analyze which areas have which needs. And one aspect of farm we didn't talk about today is we have this history of so many different field trials where we know these were the environmental conditions in that field. This is the spectral information from that field. This was the active ingredient applied. These were the microbiological needs. If we know needs were met in environmental conditions in a crop response scenario matching this novel field enrolled in B crop farm, we can predict, to use the analogy, algorithm. Watch which movies I watch with my wife and when she cuddles with me, be it because it's a scary movie or because it's a rom-com. Now predict with this movie, will my wife cuddle me when we watch this? That's what the AI recommendation engine does. It looks at your needs, it looks at your environment, and it is able to predict with confidence, okay, what tools can I bring to bear on my farm that will have me working smarter, not harder, putting resources where they're going to provide a return.
Well said. Well, thank you both for joining us today. I hope this webinar was insightful and give you a little more insights as to where things are headed, or things to consider this fall. Again, we'll have another webinar, our last of 3, next Wednesday, the 27th at 10 o'clock Eastern time. It's titled The Missing R, so it really delves into what we were kind of finishing up today of you may have the right source, the right rate, the right time, and the right place, but if you don't have the right biology, the right cyclers, the right things, moving things back and forth, you may have been missing that last step. So please tune in to next week. But again, thank you all for joining. Thank you, Lance. Thank you, Scott. And we'll let you get back to the rest of your day. Everyone have a great and safe day.
Thank you.